So my name's Joanne Wragg, and apart from being the Chief Pharmacist at Sheffield Children's, I'm also the Chief Pharmacist for the North East and Yorkshire Genomics Medicine Services Alliance. I've got with me today Emma Groves, who is our lead pharmacist for the GMSA. I think there's a slide here about you, isn't there? So Emma at the top of my screen, maybe not yours, has worked at Newcastle hospitals, and in 2016 she started down the road of the MSc course in genomic medicine, and really is a really good advocate for that particular domain of pharmacy. And she's also really interested in educational development and supporting the workforce to adopt genomics into current practice. And also joining us today, we're very excited because we thought we were going to have to try and do her tricky science part ourselves. It's Julie Atkey, who is a clinical scientist in genomics with an interest in education and training, and she is our North East and Yorkshire GMSA education and training lead. So I'm very pleased that she's coming. So for the first 5 minutes, we've got to do welcome and introductions. We've kind of done that now. Then Emma's going to talk about the genomics, getting the genomics conversations started. We did a bit of a survey, when I say we, I mean Emma, about what people want, and it was just a very basic introduction. So Julie's going to do us Genomics 101, and then Emma's going to talk a little bit about genomics in practice. During the presentation, if you have any questions, just put them in the chat and I'll keep an eye on them. And if we have time at the end, which we probably ought to have, we can do some questions and answers. So I'll pass over to Emma now.
Right, so hi everyone. Just to mention that the way we're approaching these educational learning at lunches is we're doing them in bite-sized chunks. So we'll try and communicate key bits of information in a bite-sized way because we know that everybody's so busy. So to kick off, we'll start with thinking about what is genomics. So genomics is the study of the genome, and that's the complete set of genetic material within a cell, and that forms the instruction manual for that cell to develop and function. And you might have spotted that genomics is increasingly becoming relevant within the news. So in terms of COVID-19, you may have, I just want to check, can everybody still hear me? It's kicked me out.
Yeah, I can hear you.
Oh,
but
sorry.
I've lost your slides now, though.
Oh, sorry, my computer made a funny noise and I thought it might have kicked me out of Teams, so I'll continue. But yeah, coronavirus has been a key example of how genomics is being used as part of NHS care, and it's really changed the way the patients and the public perceive genomics. Also, in terms of the NHS is really being highlighted as one of the first healthcare services to use routine genetic testing, and it will transform patient care. And I think we're at a very exciting time point where there's been significant advances in genomic technology, and we're just at that intersect where we're seeing that translate into patient care and seeing more druggable genetic targets. And finally, just to highlight that there are ethical considerations around genetics and genomics, and it's important that we have conversations with patients and the public to get this right as we mainstream genomics in the NHS. And this news article highlights discussions around the pros and cons of whole genome sequencing for babies in the UK. So let's start the genomics conversation. So what is the role of pharmacy in genomics? Now, we know that pharmacy staff in hospitals, community, primary care and mental health already make a significant contribution to the safe, effective and cost-effective prescribing of medicines across the healthcare system. And genomics is going to be another piece of that puzzle. So pharmacy teams will have a key role within precision medicine and ensuring the safe and effective and appropriate use of medicines, embedding that genomic information within decision making. Pharmacy leadership will also be absolutely key as we start to embed pharmacogenomics into mainstream medicines optimization. So pharmacogenomics looks at the genomic determinants of drug response, and it's a key area that we will focus on in future learning at lunch sessions. In terms of education and training, pharmacy professionals will also have a role in educating healthcare professionals around genomics and where it intersects with medicines and patients and the public. We will also have a role professionally having conversations with patients and making shared decision making with patients and building patient and public trust in genomics. In terms of clinical implementation, we will absolutely have a key role in developing the guidelines, prescribing and decision support around genomics and medicines, supporting with cost-effective implementation, governance and policy, and patient information. And the final piece of the puzzle, I think pharmacy teams will be also key in terms of research, and genomics is a new area of practice, and it's important that we build data and evidence which will inform future implementation as genomics moves into mainstream care. So I thought I would just quickly go through the infrastructure of genomics in England. So the Genomic Medicine Service is a world-leading initiative designed to embed genomics at scale into NHS care. And the Genomic Medicine Service incorporates 7 genomic laboratory hubs. So these are labs that are there to deliver the genomic testing in the NHS. And they deliver the testing in accordance with the National Test Directory. So on the right-hand side of the screen there, you can see a print screen of the directory. And the directory is basically like a formulary, and it outlines which tests are available in England, which patient groups are eligible, and the technology that can be used. So if this isn't something that you're familiar with, it might be worth having a look, because we anticipate that there's going to be more and more genomic tests available which impact medicines. So it's important that pharmacy teams are aware of the national directory. We also have whole genome sequencing, which is delivered in partnership with Genomics England. And on the next slide, I have an image of the new Genomic Medicine Service Alliances, which are regional infrastructures which are there to provide clinical leadership and build patient and public confidence in genomics. And they look at specific clinical pathways around mainstreaming genomics in the NHS. So in green, you can see the geography for the North East and Yorkshire GMSA. And myself as pharmacy lead, and Joanne Wragg is our chief pharmacist rep. We also have 8 pharmacy colleagues across England who we work collaboratively to think about how we can embed genomics into pharmacy practice and also mainstream medicines optimization. We have a wonderful team as well in NHSE, and Victoria Chaplin is our pharmacy genomics lead within the genomics unit. So I'll hand over now to Julie to go through some Genomics 101.
Thanks, Emma. Hi, everybody. So as Joanne said at the beginning, I'm currently the education lead for the North East and Yorkshire GMSA. Prior to that, for many years, I've been a clinical scientist in genomics. So hopefully my aim for today is to just give you a little bit of a just hopefully a refresher on some core concepts about genomics and genetics. So Emma did a brief definition of what genomics is. I think most people are probably more familiar with the term genetics, and they're interchangeable to a certain extent. But genomics really represents the kind of future direction of genetic testing. So your genome, as Emma was saying, is your complete genetic information. It includes, importantly, both the coding regions, so our genes, but also the bits of non-coding DNA in between. So genomics refers to the actual study of that genome. And just to say, there's a copy of your genome in almost every single cell of your body. So the difference between genomics and genetics is, historically, testing was around specific genes, so specific areas of the genome that code for a particular protein. Actually, within our genome, we've only got about 20,000 genes, which make up just 2% of that whole genome. So genetics refers to the study of those kind of single gene, more targeted tests. And the thing about genomics now is that by looking at both the coding regions, the genes, and the non-coding regions, we're going to establish a much better and deeper understanding of the genome and its function in healthcare. Next slide, please, Emma. So I'm sure this will just be a bit of a refresher, but in most of our cells in our bodies, not all of them, but most of them, there's a nucleus, and within the nucleus is our DNA, so our genome. And our DNA carries the instructions for the creation of our proteins, and those protein molecules are responsible for many of the functions within each of our bodies. So to get to the protein, the DNA has to go through a number of changes. So firstly, it's copied into something called messenger RNA through a process called transcription. And that messenger RNA, so that happens within the nucleus, and then outside of the nucleus in the cell cytoplasm, that messenger RNA is what we call translated into proteins. So that's the central dogma of genetics, basically, is that DNA code which is translated into functioning molecules that then perform all the different tasks within the cell that are needed. Next slide, please. So the human genome, what does it actually look like? So in most of our cells, so it's 3 billion base pairs long, and those base pairs, DNA is made up of 4 different bases, A, T, C, and G. And your DNA is a long strand of all those base pairs. But actually, if we tried to pack that enormous strand of DNA into a cell, it wouldn't fit. So what happens is the DNA is really tightly packed around different proteins and is kind of condensed into these structures called chromosomes. Within most of our cells, we have 46 chromosomes, which are 22 pairs of chromosomes plus 2 sex chromosomes. And what's happening now is we're seeing a real change in the way that we can analyse our genome. From my background historically was actually analysing chromosomes and changes in chromosomes. But the problem with that is that the level of resolution and the types of changes that we can pick up are limited. Whereas if we actually drill down and look at the actual sequence of the bases in that DNA, we can get much more information, including very small changes which might be relevant in different healthcare settings. Next slide, Emma, thank you. So when we're analysing somebody's DNA, what we're looking for is changes in the DNA in relation to what we call a reference genome, to be able to see what those changes look like, but also whether any of those changes might be responsible for the particular clinical picture within that patient. All of us have around 99.8 to 9% identical DNA. We're all human. And actually, it's quite interesting to look at the similarities our DNA has to other organisms as well. We are, the DNA is super similar to chimpanzees, as you might expect. But we're even, I think our DNA is about 60% the same as a banana. So quite interesting in terms of that kind of conservation. But the remaining 0.1 variation may contain many different variants, from very small changes, so one particular base might change, to big large changes. We can gain an extra chromosome, or we can lose particular areas within a chromosome. But not importantly, not all variants are associated with disease. There is a lot of normal variation within our genome, for example, eye colour, and drug effect, in terms of what people's response to different drugs is. So when we're talking about the types of variants that we're looking for when we're examining somebody's genome, there's two main types. So one is what we call a constitutional or germline variant. These variants arise in our gametes, so in the sperm and egg cells, and they can obviously be passed on to offspring. Whereas the somatic variant, which we see in cancer, are not heritable. They are individual specific, and so we look at those changes in a different way to the way we analyse a constitutional variant. Next slide, please, Emma. So in terms of inheritance, we all inherit half of our DNA from each of our parents, half from mum, half from dad. Each of us has our own very unique and specific genome. So even children within the same families, their genome is different, and that's because of the way that genetic material is actually inherited from each of those parents. So families look similar, but not identical, and that's why the human race is so many and varied, because of the way that genetic material is passed on. Next slide, please, Emma. So just going back to more practically then, the ways in which we can look at somebody's genome. So historically, there've been many methods developed to do this kind of analysis, and one of the biggest changes or new technologies that was introduced a good number of years ago now, but has been developing ever since, is something called next generation sequencing, and that's where you can look at many different targets all at the same time. So when we're thinking of sequencing, we can think about specific targeted sequencing, and that's where we're looking at just a small number of regions or genes within the genome, and we know that those genes are have been implicated or are associated with a particular disease. So if a patient presents with that disease, we can test for those particular areas or genes to see if there's a change within them that might be causing that patient's clinical presentation. Next slide, please, Emma. But if we want to look at more regions, we can use what's called a panel, where we can add more areas of interest, more genes for testing. And again, using this next generation sequencing technology, we can look at all of those areas at the same time. So targeted sequencing might look at a very small number of genes, maybe less than 10. Panel testing, we can look at hundreds of genes or regions all at the same time. Next slide, please. The next level of testing would be something called whole exome sequencing. Now, our exome is the coding part of our gene, so it's our 20,000 or so genes, and we can look at all of those at the same time. And then the final level, which is the most recent type of sequencing that's been introduced as an NHS test, is whole genome sequencing. So that's looking at all of the coding regions and all of the non-coding regions as well. And obviously, we're still collating masses of information by looking at people's whole genomes. The thing about each of those tests is the more areas you look at, obviously, the more data is produced, the more labour intensive those tests, and the more costly as well. Next slide, please, Emma. So that's it, kind of from a practical point of view, how we're kind of doing genomic testing. Because of this explosion in technology and the ability that we have now to interrogate people's genomes to a much greater degree, genomics is becoming much more part of different areas of healthcare, and so it's important that the workforce is prepared and educated and ready for the use of genomics as we go forwards. So back in 2014, Health Education England established something called the Genomics Education Programme, and their remit is to support the healthcare workforce to enable them to utilise genomics as effectively as possible within their role and within their practice. Next slide, please, Emma. So if you haven't already checked out the Genomics Education Programme website, I'll encourage you to do so and just kind of explore what's available. There's so much there for all different levels of learners. There's online courses, practical guidance, awareness raising through social media, all sorts of different resources, all the way through from very simple introductory resources to all the way through to an MSc in genomic medicine. And my last slide just gives you somewhere to start if you were interested. There's an 8-part course called Genomics 101 that they've developed, which starts with an introduction to genomics in healthcare and kind of takes you through testing and kind of where we are now. So that's a good place to start. Thanks, Emma. I'll pass back to you. You're on mute, Emma.
Thanks, Julie. So just to finish off, we're going to go through a whistle-stop tour of genomics and where the intersection is with personalised medicine, and thinking about where pharmacy teams will have a role both now and in the future. So in terms of personalised medicine and where we can find genomic information to inform medicine's optimisation, as Julie was saying, you can use genetic material that is found within a person, and that would be the germline DNA. We can also use information from a person's cancer, and a person's cancer genetic material is very different to their germline, and we call it somatic DNA. And the other interesting thing about cancer is that one of the reasons that this speciality has benefited so much from genomic advances is because fundamentally cancer is a genetic disease. And finally, we can find genomic information within an organism, so thinking of viruses and bacteria and how we can use that information to target treatment and understand virulence. So how is genomics enabling personalised medicine? So the personalised medicine landscape is huge, and it goes all the way from novel therapeutics, so thinking about gene therapies and the use of new technologies to really deliver tailor-made genetic material to a patient's cell to treat disease. And the examples of gene therapies include CAR T-cell therapy, which is used for B-cell lymphoma and leukemia. We also have new targeted treatments which are directly targeted to specific genomic changes. Oh, sorry, can you still hear me there? Made a little funny sound. So cancer in particular has benefited from these new targeted treatments. So in oncology, we're finding it to be more common that drugs are licensed based on their genomic target rather than the location in the body where that tumour is found. And that is really revolutionary for the field of cancer. Other areas that are benefiting from targeted treatments include cystic fibrosis, where we have drugs that target the CFTR gene. Other examples of genomics and personalised medicine is as we understand disease and the genomic determinants of disease, that also impacts treatment stratification. So for example, monogenic diabetes is a very specific form of diabetes, and it's important that we diagnose monogenic diabetes because the treatment is different. So these patients tend to respond better to oral therapy such as glycoside or lifestyle interventions. Genomics can also be very useful in determining resistance to drug therapy. So in oncology, again, we are able to sample the patient's blood. So we call it circulating tumour DNA, where you can take a sample of blood and you can look to see elements of the tumour's DNA within that blood sample, and then that information can be used to help us understand which drugs would be appropriate to target to the tumour. So it's very exciting and novel field. The other example of understanding resistance with genomics is in pathogen sequencing. So we now sequence and do whole genome sequencing for all TB cases, and then we can use that information to detect drug resistance through the genes within the bacteria. And finally, and probably one of the most exciting areas for the pharmacy profession, is pharmacogenomics. So pharmacogenomics can be used to guide treatment decisions and dosing based on genomic data, and it can be used to predict response to medicines and also adverse drug reactions. So at the moment in the UK, our approach to pharmacogenomics is quite reactive, and testing is limited to a very few specific indications. So they're kind of mainly azathioprine, abacavir, and fluoropyrimidine-based chemotherapy. However, there is evidence-based guidelines to support pharmacogenomic prescribing more commonly. So thinking of drugs like clopidogrel, tricyclic antidepressants, proton pump inhibitors, and anticoagulants all have strong evidence base for pharmacogenomics guiding dosing decisions. And I anticipate in the coming years, we're going to see increasingly more pharmacogenomic tests available via the National Test Directory. And we also need to prepare for patients as their confidence in genomics grows, that patients may be accessing private testing. And I think the pharmacy team have a key role in supporting pharmacogenomics. So, thinking of the potential pathways, there will be absolutely key roles for pharmacy teams in collaboration with all the multidisciplinary colleagues in genomics. So thinking of things like offering a genomic test to patients, interpreting results of genomic tests, returning those results to patients and contextualizing those results in the context of their disease and medicines optimization, and using genomic test results to guide management and treatment. And underpinning these pathways will absolutely be key roles in terms of finance, governance, policy, and education. So just to finish off, I know that was quite a whistle-stop tour. I wanted to flag some key resources which are more pharmacy targeted. So the Royal Pharmaceutical Society offers some great webinars around core concepts of pharmacogenomics. CPPE also have a free course around introducing genomics in pharmacy practice, and it's only 2 hours and a great starting point. There's also some bite-sized learning available via Training Matters. And finally, I would signpost you to the UK Pharmacogenomics and Stratified Medicine Network, which is a multidisciplinary network that have presentations available that are free to access from their conferences. And they're very, very good. So definitely something worth having a look at.